Okay, next up, Dr. Ashlan Reid, who's doing her postdoc at the City College of New York and looking at temporally distinct maturation of the left and right auditory cortex. Okay.
Okay. As Lena said, my name is Ashlan Reid, and I'm working at the City College of New York, and I'm so excited to talk to such an amazing, dynamic group of people. It's been really fun to meet you all. I'd like to tell you today about some of the work that I've been doing the past year and a half since we scored this Reentry Supplement Award. So I have had a non-traditional career path, and I feel like that should be championed more in academia. And, right? But I can't go into it because I'm going to run over time, because I can talk about it first, so come see me. Okay, so Lily just gave an awesome talk about brain lateralization, and lateralization of brain function is ubiquitous in the animal kingdom. We just saw how flies use lateralized behavior in courtship. Honeybees, blue whales, dolphins, elephants, and people have lateralized brain function. And what we know about lateralized brain function or the distribution from the left and the right hemispheres of these cognitive functions. A lot of the information we have about that comes from injuries to the brain, whether they're accidental or intentional in the case of epileptic patients for the treatment of seizures. So you cut the corpus callosum to keep the seizures kind of at bay. And so we have this kind of map for how the cognitive load is distributed between the hemispheres. And you can imagine how this increases your capacity to do many different kinds of tasks. But the most well-known is the parcellation of acoustic information, where you have a leftward bias of processing spoken language as opposed to other acoustic cues. And so far from being unique to humans, lateralization, one of the first studies that I did in this, I was a part of in this lab, was basically the finding that vocalization cues actually preferentially activate, if you look at FOSS expression in adult animals, vocalization sounds preferentially activate the left auditory cortex, as opposed to general sounds, which activate preferentially the right auditory cortex. I don't want to go into this data too much, but basically anywhere we see red here is a more activation in the left auditory cortex. It's kind of an index. However, the important thing is that lateralization is hearing experience dependent. So if you look at pups at hearing onset, you actually don't see any differences. And I have to apologize because this is the convention that I'm going to use for the rest of the talk. I just couldn't change the image. But for the rest of the talk, left auditory cortex will be in blue, red is always right. And so we see no difference. So something about the temporal experience that animals are having is shaping the development of lateralized brain function. And we care about this because patients with autism, schizophrenia, many other cognitive disorders are highly correlated with known dysfunction of lateralization in their brain function. So naturally, we want to be able to address this. And we've already shown that vocalizations are leftward biased. So are there any more parallels to kind of human development? Because we know a lot of different things about mouse development, and we can actually probe mouse development ethically and get at some of the kind of the mechanistic underpinnings of the development of this lateralization that's so crucial to human cognitive health. So one of these things that we want to check for is this differential maturation of the cerebral hemispheres. So the right hemisphere is known to develop earlier than the left hemisphere in people, and it's still an ongoing, a pretty hot topic. But we don't know whether that's conserved in mice. So what we do know about development in animal models, and I'll walk you through why we think time is important. And I hope to convince you of that today, that temporal differences are important in the development of lateralized brain circuits. We know that sensory experience alters neuronal circuits. Basically, this beautiful Cajal drawing, because none of the neuroscientists can avoid using a Cajal drawing in there, because it's just so awesome. But you can imagine these different cell types kind of spanning the cortical layers. So we're kind of looking at this area of the brain, really lateral. And they're intracortical and thalamocortical, so input fibers and within cortex fiber connections that are being altered by the sensory experience, so that the brain can best represent the statistics of the sensory environment. So the outside world is being translated into an internal representation. And the environment has influence over these circuits during very specific developmental time periods called critical periods. And in the mouse, this can be as short as 2 to 3 days. So we're talking about super fast time windows where if the environment is manipulated, the circuit kind of locks it into place. So it's taking this snapshot of the sensory statistics in the outside world and representing that in its circuits. And the way that, at least in the auditory cortex, information is, different parameters of the stimuli are represented in this kind of topographic gradient. So much like retinotopy in the visual field, there's tone frequency tuning across the auditory cortex. But these maps don't just pop up out of nowhere. There's a little bit of like intrinsic bias, but then it's refined over time to these circuits that basically represent the outside world. So basically, if we have a difference, a hemispheric difference in critical period timing, you're taking that snapshot of a different time of your experience in the world. So maybe your ear canal is opening, or your brothers and sisters start squeaking to you more. So over time, you're kind of viewing a different world. So what we're trying to answer is, are there time differences in maturation between the hemispheres in the mouth auditory cortex as there are resembling human development? So I'm not going to go into this in the interest of time, but let's start with the limbic-cortical axon connectivity patterns, and I'll just kind of explain each number as we go along. So thalamic input to the cortex in humans and in rodent models is known to start out in young animals going to these subplate neurons. These are the cortical layers. There's a couple missing here in this cartoon, but basically the cortex is in this laminar structure again, and the thalamocortical input is coming in and synapsing first in the young animal in these subplate neurons, and then over time, these axons move their connectivity to neurons in these upper layers. So that's what we're going to be looking at first as a hallmark of maturation. And classically, this is done in a living brain slice in vitro that preserves the functional connectivity between the thalamus and the cortex. But the problem with this is, to make this compound slice angle through the brain and keep it alive and healthy, you make a slice right through the other hemisphere, usually the right auditory cortex, because people care about the left. You make a slice through the brain to block and get this compound angle so that you preserve the connectivity. So again, these are living brain slices, so the connection between the thalamus and the cortex is alive and able to do its thing. They're acute, they're not cultured. So the first thing we did was invent a preparation that allowed us to retrieve both hemispheres, thalamocortical slices. We're going to use voltage-sensitive dyes, and I can get into the reasons why in a little bit. We're going to stimulate in the thalamus, and again, we're going to watch for responses way up here in the cortex. And what this looks like in an older left hemisphere animal, again, like the stimulating electrodes down here. So the axons course up and come into the cortex, kind of at this classical thalamocortical input location in the mature animal. And so that's one of the main reasons we're using voltage-sensitive dyes. These are, voltage-sensitive dyes span the range of activity. So as opposed to calcium indicators, these things are fast. They're on the order of milliseconds rather than hundreds of milliseconds, and they represent synaptic input rather than spiking output. So we're really interested in where this initiation happens. And now we're going to look at it across time. So I'm going to take those movies, break out the frames. The one that we just looked at was old left auditory cortex, so it's here. I've kind of marked where this kind of stereotypical boundary is for the mature thalamocortical response. And these kind of quantifications underneath it are cumulative histograms of activated pixel depths. So basically you can see that all of the pixels that are activated are below the line in the initial phases in the young animal, and they're above the line once the animal gets to be showing the mature patterning. And actually I picked, here I'm showing P12 and P18, so these animals are just 6 days apart. So this dramatic change in input location has changed over 6 days. Very similarly, and nobody knew this because they always chopped through the right auditory cortex, not surprisingly, the similar trajectory from a low to high actually occurs in the right auditory cortex as well. But if you check out, I've actually put this example in to say, anybody wants to talk about the quantification, I'm happy to talk about it, but I'm not going to go into it right now. If I chose these examples intentionally, checking P12 as before, but now we're going to only P15. So 3 days later, and the right auditory cortex is already showing this shift. And so if we look within the same animal, because now we can retrieve connected thalamocortical slices from the same animal, we can actually see that on a particular day at P16, the left auditory cortex looks immature. It shows this immature patterning, and the right auditory cortex looks like it has already shifted the axons up. This holds across the population. These are young animals here. There's no difference. The older animals have shifted, but there's this really short time period that coincides with the critical period where the right hemisphere has already shifted the thalamocortical axons up, not to anthropomorphize the cortex, but the left auditory cortex is kind of lagging behind. It has not shifted up yet, and these are 95% confidence intervals. It's pretty robust. And then this is just a single metric to tell you the same thing, which is basically right jumps during this very first kind of age range. Left stays insignificant until the later ages. So now we're going to take a look at inhibitory synaptic properties very briefly. So these are inhibition in the cortex is known to be associated with opening and closing of the critical period. So that's why we're going to do whole cell patch electrophysiology, patch on the cell, get electrical access to the insides of the cell, and look at these spontaneous inhibitory potentials as a readout of kind of inhibitory tone. And that's known to go from basically very low frequency events to kind of a more intermediate level. So again, we see this jump up in this particular kind of maturation hallmark, where we go from in the right auditory cortex to a significant shift, and then it just kind of stays the same. There's this really interesting kind of hyperconnectivity time in the left auditory cortex where you're seeing a lot more inhibitory potentials happening, or currents. So this is all great, but it's all in vitro. So what does it actually mean for in vivo? Are these differences big enough to actually make a difference? Can we alter the frequency maps differently if we basically take advantage of a tone rearing protocol where we overexpose the animal to this green 7 kilohertz tone? And if you do that during the critical period, you actually see an overrepresentation of the green frequency in the representation of the outside world, because it's present in the outside world more. So the brain commits more space to it. So we're going to do two different tone ring protocols. One where we've seen from our slice work that that the right auditory cortex is changing, but the left auditory cortex is not. And then another late rearing protocol. We're going to use multi-channel silicon probes and record spike activity in the adult animals. So we do the tone rearing as pups, wait until the animal gets older, so we know that these circuit changes have been locked into place. And these are pretty preliminary data. I literally analyzed them on Sunday night. So, but what I think they're so compelling because it really gives you a chance to see the real world, that it's actually relevant to the animal. So I hope you can see that in the control animals, you have this tonotopic gradient. There's green, there's yellow, there's orange, there's red, there's dark red. If you rear in the early, this early phase, you see a dramatic overrepresentation of the 7 kilohertz tone in the right auditory cortex, but not the left. And that's the whole idea. Then if you rear in a late auditory, so that's a pretty obvious difference. If you rear in the late time, you also get this overrepresentation in the left hemisphere, but not the right. So I hope I've convinced you that left and right auditory cortex show time differences in maturation, and these representations the representations inside of those cortices are differentially influenced by environmental manipulations. And I went and tried to do, this is a joke, I just don't deliver jokes very well. So I went and tried to do a circle, and I came up with this flower because I'm bad at making decisions, and I was trying to figure out, I'm interested in everything, so I actually think it kind of represents me a little bit. So does it have to be a circle? But it actually gave me a chance to really kind of refine. I'm okay, I gotta tone it back, you can't be crazy. And so what are the kind of common denominators here? So I've already workshopped this figure a little bit, but I'm really, really ready to workshop it some more. But anyways, I think that as an electrophysiologist, and I think I might, not to insult anybody here, but I think I'm the first person to do electrophysiology here yet, so far. But basically, I think the interface between electrical signals and molecular signals is a really understudied field where basically electrical signals can basically influence molecular signals. Molecular signals build the proteins that change the subunits of the receptors, changes the channel dynamics. Basically, the inhibitory postsynaptic currents that I showed, the metric I didn't show you was that the time constant changes, which can actually be reflective of the subunits that are being expressed. There are other mechanisms at play. But basically, you can get this loop where molecular signals then change the electrical signals because they change the channels that are happening and making these dynamic firing patterns. And so basically the underlying theme is neural communication. And I'm right at time, so I'll just briefly say this because I love it. I'm also a zebrafish fangirl. I love zebrafish, and I like everybody that's been like zebrafish. I'm like, yes. And this is the lateral line, a bundle of axons going down the side of the zebrafish and synapsing on these little tufts of hair cells that sense water motion. And if you look really carefully, this is actually kind of a little oversaturated, but if you look on the other side, you can see that there's another little lateral line. There's another bundle of axons going on the other side. So we have lateralized movement of these axons. I think zebrafish are like super awesome because they're easy to do transgenesis in. The green is actually GCamp, but since this is standard confocal, you don't really see the dynamics yet. But but I've set up with a friend now because my lab is moving St. Louis and I'm not moving with them. So as of next month, I'm going to be building a microscope, and I'm hoping we can actually test out bilateral simultaneous imaging for use in the mouse. I mean, this is left and right auditory cortex and time-lapse imaging of circuit function signal dynamics. I just thought I was running over. Anyways, so ask me about my confession time, and I'll tell you more about my interest in zebrafish. So this is the lab, a master's student, an undergrad, a graduate student, Heisel, and me. It's a small, very small lab, so I've built all of this from scratch. The VSD rig I built, the probe recordings I built, but these people are just awesome, and it's been really fun. Thank you very much.
Awesome.
Sorry for going over for a minute.
Okay. Okay. I have so many questions, but I'll try and narrow it down to a couple.
I'm good for drinks.
Yeah. Well, like we've got to chat. So one thing which, maybe is not directly of your interest, or I don't know if you have the answer to this, but I'm just so curious, so I think when I think about auditory sensory input, I think about like a labeled line, frequency maps, blah, blah, blah. I've literally never thought about the letterization thing. So where and how are different sounds being determined? Like, how do you parse between language versus music? How is that coded and then sent to each side?
I mean, right?
Yeah.
We know about tonotopic gradients, but we don't know about context gradients. I mean, it's kind of like the olfactory system where context gradients, right? Let's see if I can get back to it. Sorry.
Is it different peptides in the thalamus?
I don't know, but
okay.
I'm really excited to figure out. And in fact, kind of my pandemic, the zebrafish was a pandemic project, but it also kind of led, I took my zebrafish constructs that I was building and put them into an AAV and injected them into the LGN, the visual thalamus, and I saw a differential expression of my reporter based on which thalamic relay neuron it was in fact was expressing. So basically super brand new unknown kind of thing, whether it was getting input from the ipsilateral eye or the contralateral eye. So basically there's a region, and I was like, why is there a hole in every single injection that I do and our tech does? There's a hole of expression. So it's either boring and it's some kind of tropism, or it's really interesting, and we've come across some kind of promoter, a special promoter that basically helps the molecular mechanisms kind of jive with what's happening.
Okay, super cool. And I'll be really quick about the second
one.
So with humans, we know that, musicians, for example, have larger cortical areas dedicated to hearing, bigger auditory cortex, essentially. Mm-hmm. And so you're talking about these critical periods, but we see this plasticity in adulthood. So do you have any plan on looking?
I think innate and then refinement is super. I think I put that in. But anyways, the other thing is this Williams brain syndrome. Somebody was talking about it earlier, and I was like, I know what that is. It's when you're actually hyper aware of your.
They're hyper empathetic.
It was you.
Yeah.
So exactly. But they're really bad, dysfunctional at communication, right? So I would argue that is kind of evidence that lateralization of brain function is not really happening there. So I think it would be super interesting. And there's mouse models of this that that's
yeah.
the
Okay,
next
we
thing
need to
I want
talk.
to do.
We need to talk. I'll shut
Right.
down.
So mouse models, and then also the other interesting thing that I'm really sorry for not saying this out front. I should have. I just forgot. All of these experiments were in male mice. I had outliers. So I'm doing these pups. They're very hard to sex. So I cut their tails as I'm doing the slices, and I PCR for male, female. And so my 3 outliers were all female. So I was like, this is important and interesting on its own, but I am not made of time, and I need to like move forward with my career trajectory because it's, it needs to go. But I think that there's this world where you look at all the different things that are kind of coming together to form neural circuits. And I feel like you have to have both the molecular side of things and the activity side of things, because activity feeds into, that's how you know where you are. That's how you know what's happening for your synaptic refinement. So I'm really super excited about synaptic refinement.